video, I will speak on properties of cardiac muscle. As you all know, we have three types of muscle: skeletal muscle, cardiac muscle, and smooth muscle. Already I have delivered my lecture on properties of skeletal muscle. Almost similar type of properties also we can see with cardiac muscle, along with some differences. Some specialities we can see with the cardiac muscle if we compare with skeletal muscle or smooth muscle. Now first is excitability and contractility. Then rhythmicity. Then refractory period, then staircase phenomenon, and then all or none law. I will explain these 5 properties, not more than that. Excitability and contractility, talking about that, I would like to draw a diagram. As you all know, this is the heart, and we have SA node, AV node, the internodal pathways, then internodal pathways, this is SA node, this is AV node, then this is Buckman's bundle, then we have internodal pathways, then we have AV node, AV bundle, AV node, AV bundle. This is again left bundle of his, right bundle of his, and then Purkinje fibers. These are specialized conducting tissues in the heart. And why it is specialized, that also I have explained. The SA node can generate impulse by its own, because SA node, AV node, Purkinje fibers, these are all specialized tissues. Normally, SA node actually can generate impulse by its own, and this impulse in the form of action potential will be traveled through this internodal pathway, Buckman bundles, and right atrium and left atrium will be stimulated, muscles will be stimulated, followed by there will be stimulation of both right and left ventricle, and this stimulation will go through this bundle of his and ultimately Purkinje fibers. And again there is a junction between Purkinje fibers and the ventricular muscles. So what I want to say is the cardiac muscle has a property of excitability and conductivity. Once it is excited, these electrical impulses are conducted through this bundle, and ultimately whole cardiac muscles will be excited. So there will be a contraction followed by relaxation. So electrical event, as you all know, mechanical event will be there, and this mechanical event actually because of electrical event. This excitability and contractility, easily we can do it in the lab. If we have frog, living frog, not dead frog, living frog, then we can do this experiment easily. Say this is the board, and this board is with the stand, and as you all know, this is the frog, This is the frog. We can give an incision here and we can expose the heart. We can expose the heart. Say this is the heart. We can expose the heart, and this heart is connected with the liver, and if the kymograph is there, then we can record the systole and diastole of this heart. This is the frog, you can see from this diagram. Easily we can, after teething and stunning, we can expose the heart, and the ventricle is there. This is the apex of the ventricle. This apex of the ventricle is cannulated here. Cannulated means just a alpine has to be passed, and this alpine is connected with the thread, and ultimately it is connected to the liver. So what type of recording we will have? This type of recording we will have. This is atrial event and this is ventricular event. So atrial contraction followed by ventricular contraction and relaxation. So easily this movement this is a mechanical event, and this mechanical event we can record in the lab. So by recording this, we are going to prove the property, that is excitability and contractility. Another property is rhythmicity. Means, see here, each bit coming here, each contraction and relaxation coming with a fixed interval. Interval is same. Means, whatever the event is generated here, the electrical event is generated here, this electrical event is generated rhythmically. There is an interval, and that interval is fixed interval. Means we can say that heart has got this property, that is rhythmicity. Now coming to the refractory period. This also we can refractory period, as you all know, if we draw the curve here to talk about the refractory period, here this is the membrane millivolt, and this is the time. This is the time, and this is in millisecond. 100, 200, 300. This is 100, 200 and 300. Now if we draw the electrical event, it looks like this. It looks like this. So here we have the first 2/3 is absolute refractory period. This is absolute refractory period. And the last 1/3 is a relative refractory period. Now total duration of the action potential is 300 millisecond. The resting membrane potential is minus 90 millivolt, and this is plus 40 millivolt. So here we have depolarization phase, plateau phase, repolarization phase, and the positive after hyperpolarization phase. So these are the phases we have in action potential. And also I have marked here which one is absolute refractory period and another is relative refractory period. The first 2/3 is coming under absolute refractory period, and last 1/3 comes under relative refractory period. Now this refractory period also can be tested, as already I have explained, that if we provide second stimulus during absolute refractory period, there will be no response. But if the second stimulus, strength of the second stimulus is more than the first, and that stimulus is provided in relative refractory period, then we will have the response. So here we can test it with the help of a stimulator. Say this is the stimulator. This is the stimulator. Please, already I have drawn the diagram of the stimulator, and it has got the two electrode, one positive electrode and another is negative electrode. So if we stimulate the ventricle, if we stimulate the ventricle, then we can have an event, means extrasystole can be recorded here. Extrasystole can be recorded here easily, and by recording this, we can prove that if we provide second stimulus during absolute refractory period, heart will not respond. But if the second stimulus, strength of second stimulus is more than the first, and that stimulus is provided during relative refractory period or after relative refractory period, we will have the response. What type of curve will be there? So here, instead of this, just I am removing this. What type of curve will be there? This, then like this. See here, there is a extrasystole. This extrasystole is recorded here, and this type of response we can record, and provided the stimulus is provided after relative refractory period. So this extrasystole can be recorded by using the frog. Then staircase phenomenon is very nice experiment we can do. Staircase phenomenon. Staircase phenomenon, for that, what we have to do is, for staircase phenomenon, what we have to do is, we have to apply the ligature here. We have to apply the ligature. Ligature means, धागा तो आप जानते होंगे, सफेद वाली जो धागा होती है, उस धागा, ये आप वेंट्रिकल देख रहे हैं, इसमें we have to apply a thread here in between atria and the ventricle. What will happen? As we all know, this is the heart. So if we apply the thread here, then whatever the electrical event is generated here, it cannot be reached. It cannot be reached. This excitatory wave will be stopped because we have applied the ligature. This ligature is called first Stanius ligature. First Stanius ligature. If we apply this ligature, then see what type of record we will have. Initially we will have this type of record, but applying this, heart will be stopped for a period. So if we record this type of record will be there after applying first Stanius ligature. Now here we are supposed to talk on staircase phenomenon. For this staircase phenomenon, what we will have to do is, as soon as it is going up, as soon as stylus, which this pointer I am talking about, as soon as it is coming down, we will have to stimulate. We will have to stimulate like this. Again it is coming down, here we will have to stimulate. This is the point of stimulus. Again it is coming down, we will have to stimulate. Then again it is coming down, we will have to stimulate. So what type of recording is there? The height of contraction is increased. And this height of contraction is increased. We have not changed the strength of stimulus. What we have done, just as soon as it is coming down, we have to stimulate. As soon as it is coming down, we have to stimulate. So what is happening there, as we all know, the mechanism of skeletal muscle contraction, where calcium ion I talked about, what is the role of calcium ion? At the time of relaxation, calcium ions are sent back. But here, as soon as we are giving second stimulus, all calcium ions are not being sent back. Calcium ions are remaining there. So this calcium ion will keep adding on the response. So there will be increased number of cross bridge formation. So gradual cross bridge formation, number of cross bridge formation is increased. Due to that, the height of contraction is increased. But this type of thing we can see up to a certain limit. After that, if we stimulate, we will have same height of contraction. Although we are putting the stimulus, we are giving the stimulus, but we will have the same height of contraction. So in this experiment, we can say that there is a beneficial effect, and this beneficial effect because of the beneficial effect, we have gradual increase with the height of contraction. Gradual increase with the height of contraction, and we call it staircase phenomenon. All-on-Law law. How we can prove the all-on-Law law is a very nice experiment. The same, after putting the first stainless ligature, this type of heart will be stopped. Heart will be stopped. This type of rhythm will not be there. So what type of rhythm will be there? This type of rhythm will be there. But if we want to test the all-on-Law law, then what we have to do is all-on-Law law. After a big gap, after a gap, in case of staircase phenomenon, as soon as it was coming down, we had to stimulate. But in this case, we have to put a gap, big gap. Then, if we provide the stimulus, we will have the response. Then again it is coming back. After a big gap, again if we stimulate, there will be a response. Again there is a big gap. If we stimulate, there will be a response. But in this experiment, what is happening? Every time the height of contraction remains same. There is no gradual increase with the height of contraction. Why? Here, all the calcium ions which were released at this stage are going back, because the second stimulus we have provided after a big gap. In this gap, all calcium ions are being sent back. Again we have stimulated, calcium ions are released. So every time the number of cross bridge formation remains same. Number of cross bridge formation remains same. Here, if it is 50 cross bridge formation, here also 50 cross bridge formation are there. Here also 50 cross bridge formation there. Here also 50 cross bridge formation there. So everywhere the height of contraction remains same. Means what I want to say is, if we put a big gap after relaxation period, then we will have the same height of contraction. So what conclusion can be drawn is if heart has got 100 fibers, just as for example, then if we stimulate, 100 fibers are involved. Here also 100 fibers are involved. Here also 100 fibers are involved. So that is the all or none law. If the stimulus is not adequate, then no one will be involved. So this is the all or none principle. Easily we can do in the lab, and these 5 properties can be tested easily in the laboratory. I think all of you have understood. All of you are understood what are the properties of cardiac muscle. If I repeat again, excitability and contractility, rhythmicity, refractory period, staircase phenomenon, and all or none law. Simply sitting in the lab, we can do the experiment. Another, if you have time, you can follow this video. Effect of temperature on cardiac muscle we can test. Effect of different ions we can test on intact heart as well as on isolated heart. Just what we have to do is, just we have to keep changing the temperature. If we change the temperature, then what will happen? This is the height. But if we change the temperature, if we increase the temperature, say it is 10 degree more than the room temperature, then the height of the contraction will be increased, rate also will be increased. Height will be increased, rate will be increased. But if it is the 10 degree less than the room temperature, then the process will be slow. And the height of contraction also will be less than the initial height. So effect of temperature on normal cardiogram also can be done in the lab, and we can talk about excitability, contractility, rhythmicity. Thank you.